Okay, continuing with the cell cycle and cell division. So today we'll discuss about the mitosis. It is the stage which comes after the interphase in the cell cycle. So mitosis is a cell division which occurs only of short duration in the cell cycle when compared to the interphase. So in our previous session we came to know the detail about the interphase. So the continued part, that is the mitosis, is a type of cell division in which the parent cell undergo division to form the daughter cells. Parent cell undergo division to form the two daughter cells. And the chromosome number will be maintained. If the chromosome number in the parent, if it is diploid, the chromosome number also will be maintained same as that of the parent. If it is diploid, the chromosome number also will be 2n in the daughter cells. For example, in human beings, if the chromosome number, if it is 46, the daughter cells also, both the daughter cells will get 46 chromosomes number will be maintained. Because of that, mitosis is also called as an equational division. Means equally the chromosomes will be distributed. So this is the mitosis. The parent cell divided into two daughter cells maintaining the chromosome number. So where does the mitosis occur? Mitosis occur in the animal cells, plant cell, in all the unicellular cells also the mitosis is going to occur. Generally in animal cell or the plant cell, the mitosis occur in the somatic cells or body cells. Mitosis occurs in the somatic cells or the body cells. And generally the somatic cells and body cells will be diploid. In animals, the mitosis occur in the diploid somatic cell, but with only few exceptions. In the case of male honeybees, the mitosis occurs in the haploid somatic cells. Generally occurs in the diploid somatic cells, but only in few exceptional cases. Example, male honeybees, mitosis occur in the haploid somatic cells. That is in case of the animal. In case of the plant cell, mitosis occurs both in diploid and haploid somatic cells. In higher plants, the mitosis occurs in the diploid somatic cells. In the lower plant cells, the mitosis occurs in the haploid somatic cells. So in this way, generally, the mitosis occurs in the diploid cells, but there are also except cases in which the mitosis occurs in the haploid cells. So occurs in the somatic cells. So now let us know about the mitosis in detail. Mitosis, the cell division which we discussed in our previous session, that it has 4 stages, that is prophase, metaphase, anaphase, telophase. So let us know all about that in detail. Generally, mitosis has 2 stages which is called as an karyokinesis and cytokinesis. There are 2 important stages. When cell is fully grown up, now it will undergo interphase. So here you can see the interphase stage. So interphase, last class we came to know it as G1, S, G2 phase. All that. So here you can see this is an interphase stage. Interphase. So though you don't observe any changes in the interphase, but internally there will be an preparation will be undergoing. In the G1 phase, the components, cellular components, cytoplasmic components, all it will be synthesized. In the S phase, DNA duplication will occur. In G2 phase, again the growth of the cell and the preparation will continue. So in the interphase, though it appears to be like resting, but preparation will be undergoing. So this is the interphase where you can see that the cell membrane and here the duplicated centrioles are getting duplicated. Centrioles. In case of plant cell, centrioles are absent, and this is the nuclear membrane, chromatin network. So we know that generally the DNA, it will be in the form of an coiled thread-like structure which is called as an chromatin network. Only we can observe the chromosomes during the time of cell division. So now we can see the cell division as it proceeds, you can observe the chromosomes. So we'll come to that later. So generally when the cell is resting without any division, we have already told that DNA, it will be in the form of thread-like structure called chromatin network. So only after the division it is going to differentiate into chromosomes. So this is an coiled thread-like structure DNA, that is chromatin network, and this is the nucleolus. So this is an interphase stage in the cell cycle. Cell will prepare and you don't observe any changes. So only the chromatin network coil, it will be present in the interphase stage. So once the cell enters into the division, that is in the M phase, the karyokinesis and cytokinesis will occur. Karyokinesis means division of nucleus. Cytokinesis means division of cytoplasm. So here you can see the cytoplasm is being divided. Cytokinesis. Karyokinesis means the nucleus changes occurs in the nucleus and the nucleus is getting divided, and that is called karyokinesis. In the karyokinesis, again it is divided into 4 stages: prophase, metaphase, anaphase and telophase. In this way it has 4 stages and the changes occurs in the nucleus. Let us discuss one by one in detail. So after the interphase, that is after the G1, S and G2 phase, now the cell will enter into the prophase. So this is the first stage of the mitosis, that is the prophase. This is the prophase. So what changes will occur? Let us see in detail. Prophase, the chromatin network which is being tangled or coiled, it will become uncoiled and start condensation. So it will become condensing. The long thread-like chromatin network, it will start condensing and it will become short and thick. Okay, let me repeat. When the cell is resting in the interphase, chromatin network, long thread-like and all, it will be coiled. This chromatin network, which is coiled, it will start separating and it will start uncoiling, and then it will become condensing, which is becomes short and thick, and which is called as an chromosomes. So generally it will become chromosomes, and this is the one important changes which occurs in the prophase. So depending upon the cell, the number of chromosomes will vary. For example, in human beings, it will be of 47 chromosomes. So here we can, in order to understand it clearly, I have mentioned here 2 chromosomes. So here I have mentioned 2 chromosomes, and this chromosomes is become thick and short, and it has been condensed and it has become uncoiled from the coiled chromatin network. So from this it will changes to this when the prophase begins. But completely the chromosome still it will not be formed. The chromosome still it will be longer and thread-like. Once the chromatin network start condensing into the chromosomes will start moving towards the nuclear membrane, and the nuclear membrane, it will start disappearing. It is one of the important stage in the prophase. The nuclear membrane, it will start disappearing, and also the nucleolus also will disappear. It will also start disappearing. And next, the centrioles. In case of animal cell, the centrioles it will be duplicated. In which phase? In the S phase it will be duplicated, and that centrioles, it will start move to the opposite poles. One centriole remain in one pole. Another centriole will start moving to the opposite poles, and also the protein filaments, that is the microtubules, what we call it as a microtubules, which is called as spindle fibers. So this is an spindle fibers, will start forming. The formation of the spindle fibers will start from the centriole, and also the star-shaped cellular components will be formed. Cellular components which is star-shaped around the centrioles, which is called as an aster, also will be formed. Aster will be formed around the centrioles, and the spindle fibers will be formed from one centriole to the another centriole, and the spindle fibers formation will start. And finally, at the end of the prophase, this is beginning of the prophase. At the end of the prophase, the centrioles will move to the opposite poles, and also spindle fibers formation will be almost complete. So in this way, these are the changes which occurs in the prophase. So prophase, let me brief it once again. The chromatin network, which is called uncoil and become short and thick, which is called as an chromosomes. The nuclear membrane and the nucleolus start disappearing, and in the cytoplasm, centrioles will move from one end to the another end, and the aster formation and the spindle fibers formation will start, and at the end of the prophase it will be completed. So in this way, these are the changes which occurs in the prophase. Okay, after the prophase, the next stage is the metaphase. In the metaphase, the chromosomes which are condensed, it will lie in the center, in the equatorial plate. It is also called as an metaphase plate. Chromosomes comes and lie in the center almost, and spindle fibers formation will be completed, and one centriole will be in one pole and another centriole will be on another pole, and the spindle fibers, it will come and attach to the chromosomes. So chromosomes means exactly it will not attach to the complete chromosomes. In a cell, the chapter cell, the unit of life, we know that we know the structure of the chromosomes. The chromosomes, the two chromatids will be there attached to the centromere, and there in the centromere a protein complex will be there, and that is called kinetochore. Actually, the spindle fibers attached to the kinetochore. Here also the spindle fibers attached to the kinetochore of the chromosomes. Kinetochore of the chromosome spindle fibers attached, and in this way, in the metaphase, the spindle fibers formation will be completed. Spindle fibers attached to the chromosomes means to the kinetochore, and chromosomes lie in the equatorial plate. So the complete structure of the chromosomes is visible only in the metaphase stage. So here also the complete chromosomes will not be formed. In anaphase, the chromosomes, if the centromere is going to divide, and the chromosomes will start moving apart. And hence, in order to study the structure of the chromosome, complete structure of the chromosomes, so metaphase stage is the best stage. And here, the chromosomes lie in the equatorial plate and spindle fibers formation will be completed and the spindle fibers will be attached to the kinetochore of the chromosomes. These are the changes which occurs in the metaphase. And after the metaphase, the next stage, that is the anaphase. So prophase, metaphase, anaphase all have the stages of karyokinesis, means the nucleus has been undergoing division. The cytoplasm is not dividing. Next in the anaphase, the centromere, that is the two chromatids which is being attached, the centromere is going to divide. Centromere is going to divide, and that daughter chromosomes and after the division of the centromere, so if this is the chromosomes, centromere is going to divide, and this one daughter chromosomes, these are called daughter chromosomes now. One daughter chromosomes move to the one pole and another daughter chromosomes move to the another pole. So the same thing happens in the anaphase. The centromere divide. So centromere is going to divide and the resulting thing are called as daughter chromosomes. And one side of the cell towards the one pole, the daughter chromosomes, two daughter chromosomes are moving. And to the another side of the cell, the two daughter chromosomes are moving. So this is the important stage in which, because of this stage, only the chromosome number will be maintained. So the parent cell will be diploid. We have already told that parent cell, if it is diploid, forms the two daughter cells. The chromosome number also will be maintained. If what the chromosomes number will be present in the parents, the same number of chromosomes will be distributed to the daughter cell. So how it happens? Because of this only. Because of the division of the centromere in the chromosomes here, the two daughter chromosomes move to one pole and two daughter chromosomes move to the another pole. So because this is because of the two chromatids. And here in generally the cell only one chromatid will be present, one set of DNA. Only that is that during the DNA replication in the S phase, another set of the DNA was synthesized, that is another chromatid was synthesized. So because of that, here though the chromosomes has two chromatids, and this is the exact copy of this, and these are both are same sister chromatids, what we call, and this is the exact copy of this, and hence this move to one pole and this move to the another pole as a result. And one side of the cell receives two daughter chromosomes, and here also one side of the cell receives two daughter chromosomes because of the division of the centromere. So here the chromosomes was 2. So I have mentioned only 2 chromosomes so that we can easily understand. But in case of the different type of cell, the chromosome number will vary. It may be 46, it may be 22, it may be 24, it will vary. And here also now the chromosomes, it is moving here also 2, here also 2. And because of that only the chromosome number will be maintained in the progeny cell or the daughter cells. And in the anaphase, the centromere is going to divide and the daughter chromosomes move towards one pole and daughter chromosomes another, it will move to the another pole. And because while moving, based on the position of the centromere, the chromosomes appears in different shapes. It may be like V, or it may be like L, or it may be like rod shaped also. Different shapes will occur, that is V, L, J, and also I-like or rod shaped chromosomes will be present. That is based on the position of the centromere. These they will move. They themselves they cannot move. Actually, the spindle fibers is the one which pulls the daughter chromosomes. So spindle fibers will be attached near the centromere to the kinetochore, and it will pull the daughter chromosomes to the opposite poles. So in this way, anaphase stage is going to occur. Then after the anaphase, the last stage which occurs is the telophase. in the karyokinesis. Telophase in the karyokinesis is the last stage, means the nucleus division will complete. And here the daughter chromosomes which is being formed, it will start coiling and it will get converted into chromatin network. So we can exactly the telophase, we can say that it is the opposite to the prophase. Telophase is opposite to the prophase, means what are the changes which occurred in the prophase, exactly opposite will occur in the telophase. So now let us compare. So here the chromatin network was formed into the chromosomes, but here the chromosomes, it will get converted into the chromatin network. Here it was uncoiled, but here again the chromosomes will start coiling. And this chromosomes coils into chromatin network and this chromosomes coils into chromatin network. And the nuclear membrane and nucleolus, in the prophase it was disappeared, but in the telophase it will reappear. It will start appearing. Nuclear membrane, nucleolus. Nuclear membrane, nucleolus. Here only one nucleus. But here now we have got two daughter nuclei. So nuclear membrane, nucleolus, nuclear membrane, nucleolus will start reappearing. And centriole, one will be in this and this will remain in this portion, and one centriole towards this pole. Spindle fibers formation, here it was started in the prophase. Spindle fibers formation will end in the telophase. There will be spindle fibers formation will stop, which was the spindle fibers which was formed, it will disintegrate. It is going to degenerate. In this way, we can say that prophase is opposite to the telophase. But in prophase, only one nucleus will be present. But in the telophase, two nuclei will be present. So in this way, the nuclei is being formed. Two nuclei is being formed. That is, from one nucleus, the two nuclei is being formed. That is karyokinesis, division of the nucleus. So karyokinesis include prophase, metaphase, anaphase and telophase. Now coming to the last part of the mitosis, that is the cytokinesis, division of the cytoplasm. And we know now daughter nuclei has been formed, and other cellular components, that is like mitochondria, Golgi apparatus and cytoplasmic content, all it will divide, and some amount will be present in one cell and some amount will move to the another cell, and the cell, it is going to a deep furrow will be formed and divides into two cells. Cytoplasm will be divided and it forms the two cells. Now two daughter cells are formed and these are called as daughter cells. In this way, this is called as an cytokinesis. Cytokinesis, that is division of the cytoplasm. So in this way, the mitosis has karyokinesis and cytokinesis. So karyokinesis, division of nucleus. So one nucleus divides into two daughter nuclei, and then cytoplasm divides and forms two daughter cells. So this is the process which occurs after the interphase, that is after the G1, S, G2 phase. The cell, it will enter into the prophase and end up with the cytokinesis. In this way, from one parent cell, two daughter cell will be formed and also the chromosome number will be maintained. And this cytokinesis formation, it will vary in plants and as well as in the animals. In the animals, the cytokinesis will occur by the formation of a deep furrow. So in this way, deep furrow, it is going to pinch in this way, and as a result, two daughter cells will be formed. In case of animal cell, in case of plant cell, the cell plate will be formed. The plant cell, if we take a rectangular shaped plant cell, so generally plant cell will be rectangular shape, polygonal in shape, and the cell plate will be formed, means in this way, cell plate. small cellular components will be formed in the center. As a result, two daughter cells will be formed. Two daughter cells. And this in plant cell, the cytokinesis occurs by the formation of the cell plate. And in the plant cell also you can't observe this. Centrioles will not be present and the asters also will not be present. Directly the spindle fibers will be formed in the cytoplasm in case of the plant cells because as the centrioles are absent. So this is about the cytokinesis. In case of animal cell, deep furrow will be formed. In case of plant cells, cell plate will be formed. So this is about the cytokinesis in case of plants and as well as the animal cells. So now coming to the last part of the mitosis, that is the significance of the mitosis. Why the mitosis has to occur? In case of lower organisms, mitosis, it is the means of reproduction. So in case of lower organisms, that is single celled organisms, that too in the single cell organisms, you can see here the single cell amoeba, it is being dividing and then finally it will result in two cells. That means still two amoeba are formed. That is in single celled organism, mitosis means it is a means of reproduction. In multicellular organisms, mitosis play an important role in growth and development. A small child, it will grow into an individual, that is its journey starts from the single cell, that is the zygote. That is zygote, fusion of male gamete and female gamete forms the zygote. The zygote undergo repeated mitotic division and form the small child and growth will continue and develops into an individual. All this is because of the mitosis. So mitosis, and hence because of the chromosome number also will be maintained and exactly the cell also will be same. Because of that only the chromosome number in all the cells, it will be of same. And hence in multicellular organisms, mitosis helps in growth and development and also maintain the chromosome number. It will be equally distributed from the parents to the daughter cells. And because of that, in the human body, if you take in all the cell, will be having the same number of chromosomes. That is because of the mitosis. That is the beauty of mitosis. And also it helps in the repair and regeneration of the cell. So when you fall and some tissues and cells will be ruptured and those will be repaired and it will be regenerated. Again, after a few days, the cells will be replaced. That is because of the mitosis, and hence mitosis helps in the repair and replacing or regeneration of the cell. In this way, mitosis has its own significant role in lower organisms. It is a mode of reproduction. In multicellular organisms, that too in single cell organisms, it is a mode of reproduction. Multicellular organisms, it helps in growth and development and also in the repairing about the cells. So this is about the mitosis. So in next session we'll go with the meiosis. Thank you.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.